Today I'm going to be talking about the pancreas, liver, and a little bit over some nutrients, tell you about the function of a few vitamins, and then finish up with a couple of eating disorders. So I want to begin by telling you about the basic structure of the pancreas. So the pancreas is in the abdominal cavity behind the stomach, and the pancreas is drained by the main pancreatic duct. The main pancreatic duct joins the bile duct to form the ampulla that opens into the duodenum of the small intestine. If you follow the pancreatic duct into the pancreas, it branches repeatedly. The branches of the duct system within the pancreas are lined with epithelial cells. So there's two epithelial cells that I want to tell you about because they make the pancreatic juice. Those are the acinar cells and the duct cells. The acinar and the duct cells make a pancreatic juice. This is the digestive function of the pancreas, and it's an exocrine function. The acinar cells secrete active enzymes like pancreatic amylase, and proenzymes, or inactive enzymes or zymogens like trypsinogen. In addition to the enzymes contributed by the acinar cells to the pancreatic juice, the duct cells contribute sodium bicarbonate that helps to adjust the pH of chyme as it comes into the duodenum to 7. I'll tell you a little bit about that in just a minute. So, in addition to the digestive exocrine function of the pancreas also has an endocrine function. Now, endocrinology is the study of hormones, and an endocrine function means that the pancreas makes and secretes hormones into the blood. So scattered throughout the pancreas are little clusters of cells that kind of look like tiny islands, and they're referred to as pancreatic islets. So there's about 1 million of these islets, but despite that, they only make up about 1 to 2% of the total mass of the pancreas. So most of the pancreas consist of the cells and ducts that make the pancreatic juice. The pancreatic islets are made up of several subpopulations of cells, and these cells are designated with Greek alphabet character names. So, some of the pancreatic islet cells are known as alpha cells, and that's the Greek letter symbol for alpha. And the alpha cells of the pancreatic islets secrete a peptide hormone called glucagon. In addition to the alpha cells, there are cells called beta cells. And the beta cells of the pancreatic islets secrete a peptide hormone called insulin. We're going to find that glucagon and insulin are involved with glucose homeostasis. So the pancreas has an exocrine function where it makes a pancreatic juice that's used in digestion, and the pancreas also secretes a couple hormones from the islets, glucagon and insulin. The pancreas is said to have a dual function. One of its functions is the release of hormones. That's the endocrine function of the pancreas. And it involves the pancreatic islets, or islet cells, that make up 1 to 2% of the total mass of the pancreas. So the islet cells of the pancreas secrete two peptide hormones, insulin and glucagon, and these hormones are secreted into the blood where they travel with the blood to their target cells, and they would have effects on their target cells. The other function of the pancreas is its exocrine function. That's its digestive function. And 98% of the pancreas is involved with the formation of a pancreatic juice by the acinar and duct cells that line the branching system of ducts within the pancreas. Pancreatic juice is associated with the digestive function of the pancreas, and the pancreas typically makes over a quart of pancreatic juice each day. So the pancreatic juice contains active enzymes that we've talked about: pancreatic amylase that breaks starch down to maltose, pancreatic lipase that digests triglycerides to free fatty acids and a monoglyceride. And then you've got the inactive zymogens, or proenzymes. And they include trypsinogen, chymotrypsinogen, and procarboxypeptidase. These inactive enzymes travel with the pancreatic juice into the duodenum of the small intestine. And in the duodenum of the small intestine, these proenzymes are converted to their active form: trypsin, chymotrypsin, and carboxypeptidase. And I did mention that a key reaction that occurs in the duodenum is the conversion by a brush border enzyme in the duodenum, enterokinase, of trypsinogen to trypsin. And then trypsin catalyzes the conversion of the other two into their active form. In addition to the active enzymes and the proenzymes, there is another chemical in the pancreatic juice, and that is sodium bicarbonate. Sodium bicarbonate functions to decrease the acidity of chyme in the duodenum. Sodium bicarbonate in the pancreatic juice, when it mixes with chyme in the duodenum, will increase the pH of chyme from a pH of 1 to 2 to a pH in the duodenum of 7. And a pH of 7 is the optimal pH at which the intestinal enzymes operate. Plus, by neutralizing the pH, raising it from 2 or so to 7 in the duodenum, and it stays at that pH through the rest of the intestine. By neutralizing this pH, you also help to protect the cells that line the small intestine and the large intestine from the harsh acid of the stomach. So, the pancreatic juice, because it contains sodium bicarbonate, has an alkaline pH of about 7.5. And again, when the pancreatic juice mixes with chyme in the duodenum, it will raise the pH of chyme to somewhere around 7. So, sodium bicarbonate in the pancreatic juice splits apart into sodium ions and bicarbonate ions. We don't actually care about the sodium ions, we just care about the bicarbonate ions. The bicarbonate ions will mix with the acidic chyme in the duodenum, and the bicarbonate ions will combine with the hydrogen ions in chyme to form carbonic acid. And this reaction is going to remove hydrogen ions from chyme. Now recall that the relationship of hydrogen ions to pH is an inverse or reciprocal relationship. So as the hydrogen ion concentration goes down, the pH goes up. And the function of sodium bicarbonate in the pancreatic juice is to mix with chyme as it spurts into the duodenum and raise the pH of chyme to somewhere around 7. So just a little bit about regulation of the pancreas with respect to the pancreatic juice. Of course, we want the pancreatic juice to be created and moved into the duodenum when we are processing food. So there are triggers that allow for this timing to occur. I'm getting a piece of tape. There we go. We've talked about the cephalic and gastric reflexes, and they stimulate a digestive center in the medulla oblongata of the brainstem, and that in turn stimulates parasympathetic motor neurons, whose axons travel out to a variety of places, to include the acinar and duct cells of the pancreas, and stimulate them to secrete or make pancreatic juice. We've also got the enteroendocrine cells in the duodenum. And the enteroendocrine cells in the duodenum secrete cholecystokinin, or CCK, and secretin, or SCT. Both cholecystokinin and secretin will stimulate the pancreatic cells to make a pancreatic juice. And I do want you to know that cholecystokinin will cause the relaxation of the hepatopancreatic sphincter. That's the circle of smooth muscle cells around the ampulla, where the pancreatic duct and the bile duct join. And relaxation of these smooth muscle cells causes the hepatopancreatic sphincter to open up, allowing the bile and the pancreatic juice to flow into the small intestine. So we'll move on next and talk about the liver and the gallbladder. The liver, as you know, is a very large organ found below the diaphragm and shifted over to the right side of the abdominopelvic cavity. The liver is the second largest organ in the body after the skin. I don't think we necessarily think of our skin collectively as an organ, but it is. And the liver, as you know, is very, very large. So before we take a look at the functions of the liver, let me just review quickly the hepatic portal system. So there's two portal systems in the body. One is between the hypothalamus and the pituitary, and the other is the hepatic portal system. I know I've mentioned that the word hepatic always refers to the liver. So a portal system is a system of veins that connect two capillary beds. So here's the aorta, and recall off the midline of the aorta, the celiac trunk, superior mesenteric, and inferior mesenteric. And I'm looking here at the superior mesenteric that provides systemic arterial blood to the small and large intestine. So the superior mesenteric would branch, and those branches would enter into the walls of the small intestine and lead to capillaries within the mucosa, submucosa, and muscularis. So these would be systemic capillaries fed by the superior mesenteric in the wall of the small intestine. These systemic capillaries are drained by venules that merge to form veins, and these venules and veins are part of the hepatic portal system. Ultimately, these veins become the hepatic portal vein that goes into the liver. Within the liver, the hepatic portal vein branches into smaller and smaller veins that lead to venules that are part of the portal system that feed capillary beds in the liver. And then these capillary beds in the liver drain into venules and veins that ultimately merge to form the hepatic vein that empties into the inferior vena cava and takes that blood to the right atrium of the heart. The portal system would be the system of veins that connect capillaries in the wall of the small intestine with capillaries in the liver. So, the liver makes a solution important to digestion called bile. And bile as a solution has both an excretory as well as a digestive function. Bile is a solution made by hepatocytes in the liver, and then it travels down a system of ducts into the gallbladder outside of the liver for storage between meals. The excretory function of the bile has to do with the fact that it is involved with the secretion or excretion from the body of a couple of waste products. So we know that HDLs, or high density lipoproteins, roam the bloodstream and bind to excess cholesterol that's been secreted from cells. And the HDLs carry the cholesterol by way of the blood to the liver. The hepatocytes of the liver take up this excess cholesterol and end up putting it into the bile. The bile then travels to the small intestine, and this excess cholesterol ends up in feces. The other excretory product would be bilirubin, and because bilirubin colors solutions, it's referred to as a bile pigment. Bilirubin comes from the breakdown of hemoglobin. So there are special cells called macrophages that live in the vascular sinuses, special veins of the spleen. And they phagocytize or eat red blood cells. In other words, they take red blood cells into the inside of the macrophages. And within the inside of the macrophages, the red blood cells are broken down. A red blood cell is essentially a bag of hemoglobin. And there's over 250 million hemoglobin molecules in each red blood cell. So there's a lot of hemoglobin that has to be broken down in the body every second of the day. So the hemoglobin is broken down to globin, that's the protein component, and that then is further split to individual amino acids that would diffuse out of the macrophages into the blood and become available as a nutrient to the cells of the body. They can be reused. The hemoglobin is broken down not only to globin, but also the four heme groups are split off, and they're further broken down to their iron atoms and bilirubin. So the bilirubin would leave these macrophages in the spleen, go into the blood and be taken to the liver. Within the liver, the hepatocytes put the bilirubin into bile, and the bile ultimately ends up flowing into the small intestine, where the bilirubin ends up in the feces. The digestive function of bile, and this of course would be the digestive function then of the liver, is to add chemicals to bile called bile salts. Bile salts are emulsifiers, and emulsifiers are chemicals that when mixed with fats and oils break them down into tiny droplets. And the bile salts do this, as we've seen before, in order to increase the total surface area for lipases. to hydrolyze or break the triglycerides down to fatty acids and monoglycerides. In addition to emulsification, the bile salts will combine with fatty acids and monoglycerides to form micelles. And then the micelles, these complexes, drift over to the brush border, the microvilli of the epithelial cells that line the small intestine, where they fall apart, and well, we talked about the rest of that story before. So we're going to be looking at glucose homeostasis and the liver. First of all, just a little bit about the concept of homeostasis. So homeostasis refers to physiological mechanisms that maintain a relatively constant or stable internal environment within the body. So there are homeostatic mechanisms that regulate water balance, heart rate, blood pressure, and other operating parameters of the body. So the body must maintain glucose homeostasis because it must maintain constant levels of glucose in the blood, making glucose available to the cells of the body as a fuel substrate to make ATP. So the body must maintain blood sugar levels at all times. There are a couple of fuel molecules that cells use to make ATP. So I did want to mention that the two most important fuel molecules would be glucose and fatty acids. Glucose is particularly important because brain neurons can only use glucose to make ATP, whereas most cells of the body can use glucose, fatty acids, they can use amino acids and feed them into the Krebs cycle to make ATP by cellular respiration, and there are a few other fuel molecules that cells can use as well. So cells of our body use glucose, fatty acids, and other organic compounds during cellular respiration to make ATP. But again, I want to stress that brain neurons can only use glucose. So glucose has to be available to the cells of the body, particularly brain neurons, every second of every day. Okay, so glucose is always leaving the blood and going into cells. And as a result, if you're going to maintain stable amounts of glucose in the blood over time, you have to add glucose to the blood at the same rate that it leaves the blood. We can't eat every second of the day, and certainly we need to sleep so many hours across the night. And as a result, we need to store some of the glucose that comes in with our meals in the body as glycogen, so we can use the glycogen to provide that glucose between meals. And there's always the possibility that we could go a couple days without eating. So the glucose in excess that comes in with a meal, and when I say in excess, beyond what the cells of the body need, it is stored as glycogen. Glycogen is a glucose storage molecule. It's a polysaccharide and also structurally a polymer of glucose. So mer means to repeat, poly means many, and a polymer consists of many repeating units. The repeating unit that's bonded together to form glycogen, this would be true of starch as well, is glucose. So every glycogen molecule consists of many glucose molecules. In fact, thousands of glucose molecules in a single glycogen molecule. Glycogen in our bodies is stored in a variety of cells, but there are two primary locations where glycogen is stored. One of those would be within the over 600 skeletal muscles in the body. And the glycogen that's stored in the skeletal muscles of the body is known as muscle glycogen. And there's about 400 to 500 grams of glycogen stored in the skeletal muscles within the body. It's hard for many of us to relate to grams. So 500 grams would be roughly a pound of glycogen, which is quite a bit. Glycogen is also stored in the hepatocytes of the liver. And the liver, all by itself, stores about 100 grams of glucose as glycogen. And 100 grams would be roughly a quarter of a pound. So let's talk a little bit about the glycogen in muscle, because that glycogen can't be used to help maintain blood sugar levels or blood glucose levels over time. So the glycogen in muscles cannot be used to regulate blood glucose levels, because the muscle glycogen can only be broken down inside muscle cells to glucose. And that glucose then from muscle glycogen in skeletal muscle cells is only used by the muscle cells. So glucose can go into muscle cells and be used by muscle cells, but it can't come back out or leave muscle cells in order to regulate blood sugar levels. As a result, it's liver glycogen that acts to regulate blood glucose levels over time. The glycogen in the liver can be broken down to glucose, which then can leave liver cells and go into the blood. And the glucose leaving the liver into the blood would then circulate within the blood and be available to all cells throughout the body for their use to make ATP. So there are three pathways in the liver associated with glucose homeostasis that you need to know. One of these pathways is hepatic glycogenesis, and that's the synthesis of glycogen from glucose coming into liver cells. So the X means many, and many glucose molecules are bonded together in hepatocytes to make glycogen molecules. And the glycogen then is a storage molecule of glucose. The process of hepatic glycogenesis is stimulated by a pancreatic hormone called insulin. And I do want you to know that insulin promotes or stimulates the enzymes of glycogenesis in both the liver and actually in muscle as well. Hepatic glycogenolysis is another pathway in the liver involved with glucose homeostasis. Lysis means to split. So glycogenolysis is the breakdown of glycogen. And glycogen, when it's broken down by hepatic glycogenolysis, has glucose molecules split off it one at a time. So glycogen will break down to many glucose molecules, and then the glucose molecules that result from hepatic glycogenolysis will leave liver cells and go out into the blood, and from there be distributed to the cells of the body. The process of glycogenolysis is stimulated by glucagon. It's stimulated by epinephrine as well. The third and last of the three pathways that I want you to know is hepatic gluconeogenesis. So neo means new, genesis means to create, and this is a pathway that creates new glucose. And it does it by converting what are called non-carbohydrate compounds, like amino acids, to glucose. Hepatic gluconeogenesis is accompanied by a process that occurs simultaneously in skeletal muscle cells called proteolysis. And proteolysis is the breakdown of proteins, mostly in skeletal muscle cells. It can occur in some connective tissue cells as well. But proteolysis of muscle proteins in skeletal muscle cells, like actin and myosin and myofilaments, is going to break those proteins down to amino acids. The amino acids then leave the muscle cells, go into the blood, and travel with the blood to the liver, where the amino acids are deaminated. And that means the amine group associated with the carbon skeletons of amino acids is cleaved off. And the carbon skeleton that remains without the amine group can be remodeled into glucose molecules. So hepatic gluconeogenesis is a pathway in the liver that converts amino acids to glucose. So I wanted to go over these three pathways in the liver associated with glucose homeostasis by looking at the progression of eating a meal, processing a meal, and finally going into what's called a post-absorptive state between meals. So we're going to start with sitting down and eating a meal. The chyme forms in the stomach, but there's no absorption of nutrients across the mouth, pharynx, esophagus, and stomach. So until food moves into the small intestine, we don't enter into what's called the absorptive state. But at some point in time, as the chyme forms, over a couple-hour period, depending on how much you ate, the chyme moves into the small intestine, and nutrients then are absorbed across the epithelial cells of the small intestine into the blood, and from there distributed to the cells of the body. So, when the body enters the absorptive state, where nutrients are coming in, there are excess glucose molecules, typically in the foods we eat. And so you're bringing in more glucose at that time into the blood than the cells of the body need to make their ATP. As a result, this excess glucose coming to the liver first gets taken up by hepatocytes, and within hepatocytes, that extra glucose is built into glycogen molecules by hepatic glycogenesis. So hepatic glycogenesis is the storage of excess glucose coming in with diet into glycogen. And the glycogen is a storage form of glucose in the liver. And this is a process that would be stimulated by a pancreatic peptide hormone called insulin. So, now we're going to go between meals. So, let's say that this post-absorptive state, is going to start somewhere around 3 to 5 hours or so after you've eaten a meal. And the assumption here is that you've processed all the food in the intestine, and there are no more nutrients as a result coming in from the intestine. So the intestine cannot serve at this point during the post-absorptive state as a source of glucose. This is a point where the pathway in the liver called hepatic glycogenolysis kicks in. And the liver stores, about a quarter pound or so of glycogen, around 100 grams. And that amount of glycogen can provide glucose by way of hepatic glycogenolysis for probably up to 12 hours, depending on whether you exercise. So if you're exercising a lot, maybe you only got 3 or 4 hours of glycogen in the liver that could provide glucose. And if you're not exercising at all, maybe you have up to 8, 10, maybe even 12 hours of glycogen that can provide glucose. So hepatic glycogenolysis kicks in the post-absorptive state to provide glucose from the liver into the blood, and the blood then would deliver the glucose to all cells of the body. This process of hepatic glycogenolysis is stimulated by a peptide hormone from the pancreas called glucagon. It's also stimulated by epinephrine. So we're going to assume that for whatever reason, and it may be a decision on your part, or you just can't get to any food, we're going to assume that you're in a prolonged post-absorptive state, which I'm referring to as a fasting state. And we're going to assume that you haven't eaten for 12 or more hours. So by that point, there's certainly no glucose in the small intestine coming into the body. So the small intestine isn't a source of glucose. And of course, you're not eating and adding more. And we're going to assume that you've completely exhausted or depleted the supply of glucose stored as glycogen in the liver. So there's no glucose coming in from the small intestine, and there's no more glycogen to supply glucose in the liver. But cells need glucose every second of the day in order to make ATP. And this is particularly true of brain neurons, because brain neurons can only use glucose as a fuel substrate to make ATP. So, this is when hepatic gluconeogenesis, along with muscle proteolysis, begins. So proteolysis occurs as proteins in muscle cells, and these would be the proteins in the myofilaments that make the myofibril. And the muscle proteins are broken down to amino acids. That's the lysis, or splitting apart of proteins, called proteolysis. The amino acids that result in muscle cells then go into the blood, and the amino acids in the blood travel to the liver and go into hepatocytes. And then within the liver, the amino acids are converted to glucose as their amine groups come off as ammonia. The process of converting non-carbohydrate compounds like amino acids in the liver to glucose is called hepatic gluconeogenesis. The glucose molecules that the liver makes in this fasting state then leave the liver and go into the blood, where that glucose is available to the cells of the body. Now, this is a process that cannibalizes, essentially, or eats the proteins of your muscle cells in order to supply glucose to the cells of the body. And if you continue to do this, because you don't eat for days, it will cause muscle wasting. In other words, it will significantly weaken your muscles by breaking down the contractile proteins that these muscles depend on. So the solution to the fasting state is either eat a meal and go back to where we started, or starve to death. During the fasting state, the process of hepatic gluconeogenesis is stimulated by a hormone from the adrenal gland called cortisol. So we're going to move on to talk about some other functions of the liver, to include urea formation. So there are a number of nitrogen waste products that we'll talk about. And these are chemicals that the body has to get rid of that contain nitrogen. And as a result, they're referred to as nitrogen-containing waste products or nitrogen waste products. And they include urea and ammonia, both of which are toxic to cells. And so the body has to get rid of them. So the cells of the body use amino acids for things other than building their proteins. Some of the amino acids in the cells of the body are deaminated. And that is cleaving off the amine group of the amino acid to ammonia. When amino acids are deaminated, their carbon skeletons can be refashioned into other compounds that cells can use. For example, amino acids can be deaminated and fed into the Krebs cycle to make ATP. The ammonia that results from deamination is very toxic to cells. And as a result, the body has to get rid of it. So the ammonia that cells make leaves cells and goes into the blood, and the blood carries the ammonia to hepatocytes of the liver. And the hepatocytes in the liver combine ammonia with carbon dioxide to make urea. The urea then leaves the liver cells and goes into the blood, and the blood carries the urea to the kidney, where the kidney puts urea into urine that ultimately is urinated out of the body. Okay, so we're going to look at a couple other functions of the liver to include the synthesis of plasma proteins. So there's a couple plasma proteins that we've already talked about to include albumin that helps to maintain the osmotic pressure of plasma, and fibrinogen and prothrombin that are necessary for the clotting of blood. We've already talked about these plasma proteins, but I would like you to remember those or know those for the upcoming test. So there's a couple diseases of the liver that I want you to know, and they include hepatitis and cirrhosis. So hepatitis is inflammation caused by tissue damage to the liver. And when you have tissue damage, you have dead and dying cells. So the most common causes of hepatitis would be alcohol abuse. So this would be associated with an alcoholic who's consuming a lot of alcohol every day. Alcohol is toxic to liver cells, and over time will kill them and compromise the ability of the liver to function properly. There are also some viruses that get into the body and infect liver cells, and they too can kill off liver cells and lead to a type of viral hepatitis. Cirrhosis is a chronic or long-term damage to the liver that results in dead and dying cells. And these dead and dying cells are replaced, in the case of cirrhosis, with what's called scar tissue. And scar tissue is dense connective tissue in locations where it's not normally found. So as the cells of the liver die during cirrhosis faster than they can be replaced, those dead areas are replaced by scar tissue. And that results in a permanent, irreversible loss of liver function. So there's a couple causes of cirrhosis, and they include alcoholic cirrhosis as well as hepatitis C cirrhosis, where hepatitis C is a type of virus. So just a little bit about what are called gallstones. Gallstones are rock-like crystals. So they'd be like little crystals of salt, really. I mean, just in the way they looked. And they're very hard. They're made of cholesterol, bilirubin, and calcium salts like calcium phosphate. They form inside the gallbladder as well as in the cystic and the bile ducts. The problem with these crystal rock-like gallstones, which can be as large as a 1/4 inch in size, is that they rub against and irritate the lining of the gallbladder, and that can be very painful. They can also block the bile duct, which would lead to obstructive jaundice. You'd expect that the movement of bile is compromised, and that can result in problems digesting fat. So the treatment for gallstones includes drugs that you can take that dissolve away these crystals, and that can resolve the gallstones. If that doesn't work, you might have to surgically remove the gallbladder, and you live life without a gallbladder. And a third option would be lithotripsy, which is when you pass high frequency sound waves through the body, and the high frequency sound waves will cause the crystals to shatter apart into almost little fragments that are about the size of grains of sand, and they would pass out then with the bile, and you could eliminate the gallstones that way. I'm going to finish up by talking about a couple eating disorders and then a few vitamins. So there's a couple eating disorders that most people know about. One is anorexia, the other bulimia. Anorexia is severe weight loss that is self-induced. In other words, you make a decision, essentially, not to eat so that you can lose a lot of weight and be very, very skinny. This kind of severe weight loss can lead to starvation and death. Generally, those with anorexia who practice this self-starvation to lose weight and be thin are young single females who are obsessed with weight loss because in general they have a poor self-concept. So the idea is if being thin is good, then being thinner is even better, and that takes on a life of its own. So the other one is bulimia, and that is binge purge syndrome. And that's when someone overeats, binges on food, and then vomits the food out, because you can induce vomiting by sticking your finger down into the back of your throat, or takes laxatives to encourage bowel movements in order to get rid of or purge the food they ate out of their body. And it's a binge purge that occurs several times a week. So again, the treatment is psychotherapy. This would be seeking the help of a professional just to figure out what sort of deep-seated stuff is going on that is causing you to harm your body. And of course, diet control is associated with both of these conditions. There's several vitamins that we've talked about in AMP1 or AMP2, and I would like you to have a sense of what these vitamins do. They include vitamin A, which is used by rod cells in the retina to synthesize rhodopsin. And rhodopsin is necessary for rod cells to form images in low light conditions. Vitamin D is essential for the absorption of calcium from the small intestine, and the calcium that comes into the body is necessary for the formation of the bony matrix, the hardness of teeth. Calcium ions are central to muscle contraction, and a number of enzymes have to bind to calcium ions before they become active. So vitamin D, that leads to the absorption of calcium, allows the body to build bones and function properly. Vitamin K is necessary for the synthesis of prothrombin in the liver, and of course prothrombin is associated with blood clotting. Vitamin B12 is needed to make red blood cells, and finally vitamin C is necessary for the synthesis of collagen fibers. And collagen fibers are fibers that give strength to connective tissues. Okay, well, we're done with digestive system, and this allows us to move on to our next lecture test.